Hello students. Today we shall discuss the secondary growth in Dicot stem, and I have taken Tridax as an example. So this is Rekha, working as Associate Professor in the Department of Botany and PG Studies, Bangalore. So growth, what is growth? Increase in the length is called as the primary growth. Increase in the girth is called as the secondary growth. So primary growth is due to the activity of the apical meristems. So in a plant, it grows continuously in length and stops or ceases its growth. And this increase in length of the plant is called as the primary growth. Increase in the thickness or the diameter or girth of the plant is called as the secondary growth. Primary growth is due to the activity of the apical meristem. Secondary growth is due to the activity of the lateral meristems, namely the cambium and Cork Cambium, otherwise called as Fellowderm. So secondary growth can be defined as an increase in thickness or girth of the stem and root axis due to the addition of secondary tissues by the activity of lateral meristems in the stellar and the extrastellar regions respectively. So this can be studied under two headings. One is stellar secondary growth and the other one is extrastellar secondary growth. So first let us study the stellar secondary growth. So stellar secondary growth is brought about by the Vascular Cambium. So Vascular Cambium, as the name itself indicates, it is present in the vascular bundle. It is present in between the primary xylem and primary phloem. So you can see the vascular bundle here. So in between the xylem and phloem, you can see this fascicular or intravascular cambium. So the vascular bundles in this diagram, you can see they are arranged in the form of a ring surrounding the pith region, each vascular bundle is separated from the other by pith rays or medullary rays. So how the first step in secondary growth is the formation of cambium ring. The fascicular cambium becomes active and meristematic. At the same time, so in the second diagram, you can see a complete ring of cambium. A new strip of meristematic cells arise from the parenchyma cells of the pith rays lying in line with the fascicular cambium called the interfascicular cambium. So now the inter and the intra together, they combine together and they form a complete cambial ring. So this cambium, this circular ring of cambium, it gives off the secondary vascular tissues in the stellar region. So it cuts off two types of cells, the fusiform initials, which gives rise to the secondary xylem, and the ray initials, which gives rise to the medullary rays. So the second step is how this secondary xylem and secondary phloem is formed. So the cells of this cambial ring, it divides tangentially and give rise to the vascular tissues on both the sides of the ring. The cells which are cut off towards the inner side of the cambium, it gets differentiated into secondary xylem, and those towards the outer side, it forms the secondary phloem. Thus the secondary xylem is laid down on the outer side of the primary xylem, and similarly the secondary phloem is added on to the inner side of the primary phloem. So next coming to the secondary xylem, which is also called as wood, how this secondary xylem is formed. The amount of secondary xylem formed is greater than that of the secondary phloem. The primary xylem is pushed inwards and appears as conical protuberances in the pith. The secondary xylem shows clear axial and radial system. So the axial system consists of vertical rows of cells forming tracheids, vessels, fibers and parenchyma. The radial or horizontal system is composed of parenchyma cells forming the xylem or the wood race or secondary medullary race. So next is how the secondary phloem is formed. See, it pushes off the primary phloem outwards and usually it gets crushed, losing its function. Secondary phloem takes over its function. It is made up of sieve tubes, sieve cells, companion cells, phloem fibers and phloem parenchyma. The phloem fibers constitutes the hard bast or the bast fibers. The remaining elements are known as soft bast. The bast fibers are of commercial importance and it is used as fibers in textile industry. So next one is the formation of secondary medullary rays. So these secondary medullary rays arise from the ray initials of the cambium. So the portion of the rays confined to the xylem is called as the xylem rays and which lies in the phloem is known as the phloem rays. Next one is the annual rings. The activity of cambium depends on the climatic conditions. So in temperate regions, in cold countries, during spring, cambium forms xylem vessels with wide cavities and this is called as the spring wood or the early wood. In autumn or summer, the cambial activity slows down and the xylem vessels formed are more dense and composed of compact lignified elements called autumn wood or late wood. So these spring wood and the autumn wood together appear in the form of distinct concentric circles called as the growth rings and these two kinds of wood together form the annual ring which represents the total. You can see in the diagram, you can see a number of rings, you can zoom it and see. So since one ring is formed in each year, the age of the plant can approximately determine by counting the number of rings. So next one is heartwood and the sapwood. See here in the diagram you can see two different, this is a transfer section where you can see two different color of the wood. See, as the age of the tree increases, the inner part of the secondary wood transforms into hardwood as it is present in the middle. It loses the power of conduction and it gets filled with the oils, gums, resins, tannins and other complex organic compounds. So this region becomes very hard and appears darker in color, hence it is called as heartwood or duramen, which gives mechanical strength to the plant. The outer region of the secondary wood consists of the young xylem cells and this region forms the sapwood or the alburnum. It serves the function of conduction of water and mineral salts. So this is about the stellar secondary growth. So now we shall discuss the extrastellar secondary growth. It is also known as cortical secondary growth and takes place by the activity of the secondary lateral meristem which is called as the Cork Cambium or Fellowderm. So in the diagram you can see the Cork Cambium or Fellowderm. So as a secondary growth is proceeding in the vasculature, secondary growth also takes place in the cortex resulting in the formation of the periderm tissue. The tissue replaces the epidermis and offers protection through the stem. So the periderm means the outer bark of the plant. It consists of three regions, namely the Fellowderm or the Cork Cambium, phloem or the cork tissue, Fellowderm or the secondary cortex. So you can see the phloem, Fellowderm and Fellowderm. So Cork Cambium is called as Fellowderm. The tissues which are cut off towards the inner side is called as the secondary cortex or Fellowderm. The tissues which are present outside the Cork Cambium is called as cork or phloem. So first coming to the Cork Cambium or Fellowderm. So this is a meristematic tissue arising in the outer layers of the hypodermal cells of the cortex. The cells of the Cork Cambium, they are rectangular. So they are arranged in the form of bricks, rectangular, and it produces the tissue towards the outer and the inner side. So the tissue which are produced towards the outer side, it is called as the phloem or the cork, whereas those towards the inner side, it is called as the secondary cortex, or it is also called as Fellowderm. So what is this phloem or cork? So the cells which are cut off towards the outer side by the cork cambium is called as cork or phloem. It acts as a protective layer, and more of phloem is formed from the phellogen than the Fellowderm. That means the amount of cork formed is more when compared to the Fellowderm, and these cells are dead cells. The cork cells are dead cells. They are arranged in radial rows, and at maturity their walls are suberized so that they become impermeable. So the commercial bottle cork, usually we use a cork to close the bottles, and which is called as the bottle cork. It is obtained from the plant called Quercus suber. So next one is Fellowderm or secondary cortex. So this consists of the cells cut off from the phellogen on the inner side, which differentiates into a group of parenchyma. So these cells usually develop chloroplasts and perform the function of photosynthesis, and they also serve as the storage cells. Next one is the periderm. So what is periderm? Periderm refers to a group of protective cells of the extrastellar region formed by the activity of cork cambium. So it consists of three kinds of tissues, namely the phellogen, phloem and Fellowderm. So you can see this, the transverse section of the Dicot stem showing the secondary growth. So there you can see the springwood, autumnwood, the secondary tissues, the phloem, Fellowderm, phloem, phellogen, etc. So next, coming to the bark. The bark is the region of the stem outside the cork cambium and consists of the dead cells. A few inner cells of the bark may be living. It includes several kinds of tissues including periderm and the secondary phloem. There are two types of bark, namely the scaly bark and the ring bark. Scaly bark is seen in separate, it comes out in the form of separate strips in plants. Example is eucalyptus and guava plants. Ring bark occurs in the form of complete ring. Example is Clematis, Betula, etc. So what is the function of this bark? So the function of this bark is, firstly, it gives protection to the inner living tissues. Secondly, it prevents the evaporation of water. Thirdly, the checking of invasion of the living tissues by pathogenic bacteria and fungi. And fourthly, it is the one which replaces the old cells by the new ones. So next, coming to the lenticels. So what is lenticel? So these are the epidermal apertures formed by the activity of the cork cambium. So you can see the porous tissue in the diagram consisting of the cells with large intercellular spaces in the periderm of the secondary thickened tissues, and this is called as the complementary cells, and it provides a pathway for the exchange of gases between the internal tissues and the atmosphere through the bark, which is otherwise impermeable to gases. So this is the overall summary of the secondary growth. So the cambium, you have two types of cambium. One is vascular cambium and the other one is cork cambium. So this vascular KBM, it produces two initials. One is fusiform initials and the ray initials. The fusiform initial, it gives rise to the secondary xylem and the secondary phloem, whereas the ray initials, it gives rise to the vascular rays, namely the xylem rays and the phloem rays. So coming to the cork cambium or the phellogen, so it gives rise to the periderm, which includes the phloem, Fellowderm and Fellowgen. So thank you for listening.